Good afternoon, friends. Today we are going to discuss the photosynthesis, its light reaction in very detail. That how this light reaction is important for the photosynthesis to accomplish. That means in the last video we have seen that the light reaction basically provides the chemical energy so that the sugars can be synthesized from the dark reaction, the so-called dark reaction. Though we know very well that the dark reaction is not really the dark reaction because its enzymes are light dependent. So, in a length detail, we are going to consider the so-called light reaction in which we have already discussed the Z scheme. That Z scheme would be discussed in very detail for the sake of electron transport, which is of two types, non-cyclic and the cyclic electron transport. The non-cyclic electron flow actually creates the Z scheme, although the cyclic one doesn't create the Z scheme. Now, what kind of herbicides are there which inhibits this light reaction, and at which place they are creating any kind of inhibition? So all these things would be discussed, how the ATP is synthesized, and what is photophosphorylation, what is PMF, that is the so-called proton motif. All these things we will discuss in this particular slides. So we are going to start with the non-cyclic electron transport. What happens? We know very well that there are two photosystems. And in these photosystems, there are reaction centers. For example, for the photosystem second, there is the reaction center P680, which is a reaction center chlorophyll molecule. What is the meaning of the reaction center? That all the pigment molecules would capture the light and ultimately funnel that energy into this P680, that means into the reaction center. And because of that energy, the electron of this P680 would be excited, and that would be donated to the electron acceptor, and there would be a chain of electron acceptor. And finally, all those electrons would be channelized for the synthesis of NADPH. That is the meaning of this electron transport chain. Whatever happened with this P680, that also happens with the P700, which is the reaction center of photosystem first. So what happens in a simple language, we can say, in the very first step, light excite the electron from P680 as well as from P700. And these excited electrons would be donated to pheophytene and A0, that is a kind of chlorophyll. Okay. Now, there is a lack of one electron, and there is a lack of one electron here. That means P680 as well as P700 both are get oxidized. They are not saturated with electron. They are a single electron demanding. Now what happens? One electron would be channelized from the water oxidation to P680, and then it would be funneled into a Z alphabet kind of situation like that. So one electron, due to the water oxidation, is funneled to protein D1 tyrosine, that is the YZ, then to the P680. So its electron would be saturated now. Now whatever the electron has already given to that pheophytene, that would be further channelized into quinine and plastoquinones and quinols, then finally to cytochrome B6F complex, in which many are the iron sulfur risky proteins. From this complex, the electron is taken by plastocyanin and that plastocyanin, reduced plastocyanin, then move within the thylakoid membrane. That means it's a movable protein within the thylakoid membrane and get attached to the oxidized P700. So the electron is finally given to P700. Now the electron that has already given to the A naught, that is a kind of chlorophyll, is then channelized to quinone A1, then a series of iron sulfur proteins, then to the ferredoxy. From the ferredoxy, electron is given to NADPH through FNR, that is the ferredoxine NADP reductase enzyme. So this is how the NADPH is synthesized. Now we need to understand that one. Again, you can consider that photon absorption by P680 and P700, both are get transferred, and P680, the electron from P680 is given to the pheophyte. That means P680 is oxidized and it is re-reduced by the YZ that has received electron from outer oxidation. Now this pheophyte gives electron to plastoquinones, then cytochrome B6F, and finally electron comes through plastocyanin to the oxidized P700. And similarly, the electron that has been excited from the P700 to the A naught is finally given to the ferredoxine, and that is to be utilized for the synthesis of NADPH. So that was the overview that we have considered. Now in a different kind of diagram, we will see the different story. While the electron is given from water to the synthesis of NADPH, what happens? Electron has to move from the different kind of complexes. That is the photosystem second, cytochrome B6F, plastoquinones, plastocyanins, and photosystem first. While electron is moving, these proteins or these complexes are pumping the proton ions from stroma to the thylakoid's lumen. For example, from the photosystem second, due to the water oxidation, oxygen as well as protons are liberated towards the lumen side. Similarly, due to this quinone plastoquinone cycle, cytochrome B6F liberates proton towards the lumen. So the lumen proton concentration is getting high, while at the stromal side, because the NADPH synthesis requires the proton along with this NADP plus an electron, the stromal pH is getting low. That means the free protons numbers are being reduced while lumen side proton numbers are increased. So there would be a creation of electrochemical potential gradient. This gradient is utilized for the synthesis of ATP through a protein complex called ATP synthase. The protons move back to the stroma. Downhill movement this is actually, and this cause the rotation in this particular protein called ATP synthase, and that generates the energy for the phosphorylation of ADP or for the synthesis of ATP. So this is the way how the ATP is being synthesized, and such kind of procedure is called phosphorylation, photophosphorylation, because ultimately it is the light which is considered responsible for the synthesis of ATP. That's why it is called photophosphorylation. So what happens? In this diagram, you can easily see that these protons are higher in one side of membrane and lower in one side of membrane, and this is actually creating a kind of force which drive the ATP synthesis. Such force is called proton motive force. So here we can see whatever we have discussed earlier. For the sake of revision, the transfer of electrons and the protons in the thylakoid membrane carried out by 4 protein complex. Photosystem second, then for the synthesis of NADPH consumed from the stromal site, then protons are channelized towards the lumen by cytochrome B6F, and photosystem first. So that already we have discussed in that diagram. Now we are going to consider the cyclic electron flow. By this phosphorylation mechanism, or you can say generation of PMF, we have completed our discussion of non-cyclic electron flow, and now we are moving to the next one, cyclic electron flow. What happens in the cyclic electron flow? It involves only single photosystem, that is photosystem first. The electron excited from this P700 is ultimately given to the P700 by means of this series of proteins or proteins involved. What happens here, the excited electron is not given to the synthesis of NADPH, rather than from the ferredoxin it is again channelized to the cytochrome B6F. So this situation just create the PMF, and that is to be utilized for the synthesis of ATP. Less energy is synthesized here, high energy is not synthesized, no NADP is being synthesized. So it actually happens under a certain specific condition, and such kind of energy is basically utilized or getting important for the C4 plants, where the organic acid C4 and the C3 has to move among the mesophyll and the bundle sheet, which requires extra energy. So the importance of the cyclic electron flow is basically considered for the C4 plants, that we will see later on. Next, photophosphorylation. That we have already discussed, that the Horner and the Frankel, they have demonstrated the photophosphorylation, and it was considered that photophosphorylation is nothing but the light dependent ATP synthesis, and this ATP synthesis is basically working on the model chemiosmotic mechanism, which was given by the Peter Michael for the oxidative phosphorylation, actually, in which he proposed that it is the unidirectional transmembrane proton flow. That means at one side of the membrane proton concentration is higher, and the another side of membrane there is lower proton concentration, and this generation of PM, proton motive force, is actually drive the ATP synthesis. So the same chemiosmotic mechanism is considered for the photophosphorylation. Now, the detailed study of the ATP synthesis, it is also called the CF0 and CF1. The CF0 is a part of ATP synthase, which is a transmembrane channel. That is, it is hydrophobic in nature, through which the proton can pass. And CF1 is actually the stromal side complex part of ATP synthase, which is synthesized by alternative alpha beta polypeptides, and it is the beta polypeptide which have the catalytic active site. What happens when the protons are moved back from lumen to stroma? That internal stack, which is mainly being synthesized from CF0 and partly of CF1, rotates, and this rotation is ultimately creating the energy for the phosphorylation of ADP. So at the stromal side there would be the synthesis of ATP. Now finally we are going to consider that in this light reaction, is there any herbicide which act? Yes, DCMU is the major herbicide that basically act on the plastoquinone binding site. Similarly, there is a paraquat, and that paraquat is basically act on the early electron acceptor of the photosystem first. That means A0, A1, that is basically inhibited because paraquat divert electron towards itself. So there would be no electron flow for the synthesis of NADPH for the FNR2 act, that is the ferredoxin NADP reductase protein. So these are the things that we have considered. Thank you so much. Stay blessed, stay safe.